think I'll do a DVL reset for what it's worth. Pretty sure you're up here somewhere.
Copy.
It's a guess. Taylor, and you might want to mark that just in case we're doing a DVL reset. Sure. All right, Chad, we're held here. I think as it was handed over to me, not sure if you want to still keep moving east or if you want to head maybe 045 northeast.
Yes, let's start northeast for a small move and then head north after that to kind of complete this this circle.
Roger that. Looks like I'll do 50, and then let me call that in. Bridge nav. Let's do 50 meters, 045. Thank you. And the idea here, from what I understand, and correct me if I'm wrong, is that we'll kind of try to keep the ship going and limited stopping until we reach the full circle.
Correct, yeah, so we'll just keep headed in that direction. We'll try to complete the full circle, and then after that, we'll do a tighter circle.
Okay.
In the middle, and I know that'll be smaller ship moves and stops. And hopefully we can kind of reasonably cover most of the area in maybe 3 of those circles, and then finish it off with kind of crisscrossing across the top. If you recall last year, it was relatively flat, where, you know, if it's a small area, we can just kind of mow the lawn a little bit, go back and forth. But the idea here is we definitely stopped a couple times here and there, but the priority is to try and, for myself, kind of feel like we reasonably covered most of the area for visual observation, so we can do a minimum population count at a future time. If we have any built-in time, then we can stop and do, we just did 2 measurements, optode measurements so far, and then be able to jet on over to the bump on the seafloor and the whale fall. To me, ahead of schedule, ahead of what's planned, so we can build in some of that buffer time.
Sure.
So we can make sure we get that second dive in at the garden.
Sounds good.
It's hard. We see these things and we're like, oh, I want to stop, but
yeah,
it's like, got to keep moving.
Oh, there's one of those worms. It looks like straight ahead, center of the screen. Ribbon worm? Is that the name?
This one?
Yeah.
Acorn worm.
Acorn worm. That's it. Acorn worm. Have you been seeing those?
Yeah.
Cool.
So are we still in the middle of a shift change with the pilots?
We're here now, ready to go.
All right. Hi, everybody.
Hello.
Good morning.
Hello.
So we just performed a shift change. We exchanged different pilots and navigators, video and data loggers, and a few scientists changed seats. Chad King is still fearlessly leading the pack here.
Pulling an all-nighter.
Pulling an all-nighter. How can you resist? So this is Erica Burton. I'm a scientist on shore, going through telepresence tonight. I work at the Monterey Bay National Marine Sanctuary. And Kirsten, can you introduce yourself?
Sure. Good evening. This is Kirsten Lindquist, also a scientist ashore, here assisting Erica. I am from the Greater Farallones Association and work very closely with the Greater Farallones National Marine Sanctuary research team. I'm totally stoked to be here right now.
And how about the ROV pilots, navigator, video logger? And data logger, can you introduce yourselves?
Hi, so I'm the data logger. My name is Taylor Ann, and I'm back here in the back road with Chad, taking notes of what we see, logging our temperature, oxygen saturation and concentration, and yeah, just making sure that we're getting everything that we see logged and stored in the computers so that we can share with other scientists.
This is Renato Kane. I'll be the navigator on this watch.
Dan, I'm Herc pilot.
This is Jake. I am the Argus pilot.
And this is Stephen Matter, in the video chair.
And Chad, can you introduce yourself? We've been here a few hours.
Yeah, Chad King, Chief Scientist for this cruise from Monterey Bay National Marine Sanctuary. I work with Erica on the research team.
So
And,
Chad, can you describe what we're doing?
yes, absolutely. So we are here near Davidson Seamount, a few miles off the base of the seamount on this small volcanic cone. It's probably about 200 to 250 meters in height, where last year we discovered a second octopus nursery. It's about 10 kilometers away from the original one we discovered in 2018. So this is an important site, but we didn't get to characterize it because we found it in the last hour of the dive last year. So what we're doing now is we've noticed a pattern here where all of the octopus are concentrated near the top of this feature. And if you think of, you know, you go up into the mountains and the trees will disappear. They call that the tree line. There's a little bit of an octopus line here where they start at a fairly consistent depth around the top of this really fairly symmetrical cone. And so we're kind of on the that octopus tree line, if you will, and we're about to complete the last quarter of that first circle. And then once we do that, we'll do successive tighter circles, maybe another 2 or so, and then fill in the gaps at the very top. And the reason we're doing that is because we want to understand how many octopus are here, so we can go back later with the video and count the individual octopus. It won't give us the total, but it'll give us a minimum number, so we have a reasonable estimate. If we count, let's say 1,000 or 2,000, we know there's at least that many. Normally we stop and smell the roses as we look at these anemones. I don't know, that popped in my head a little more, but we do have a weather system moving in. So we're trying to expedite this dive a bit. So we're not going to be sampling as much and doing as much zooms, and we also will periodically do oxygen and temperature measurements. We have an instrument that the manipulator can grab and stick into cracks and crevices where we see warm water coming out. And that is a key feature here. These octopus who are brooding, as if you've just joined us, we will, I guarantee you, we will come across more groups of these octopuses lined up on features. In the lines into pools and cracks and crevices, there's a clear association with the warm water. And you'll see this shimmering like a mirage or heat coming off the pavement on a hot day. And we'll put the optoad in there periodically to get temperatures. Normally it's near freezing, about 1.6 degrees Celsius is what the temperature generally is. Right now, yeah, it's 1.6 or so. We've gotten 2 temperature readings today, the highest one being I believe it was 9.4 degrees. Yes, 9.4 degrees C, which is about 49 degrees Fahrenheit. That's like a hot tub for these guys, even though it's still cold for us. But yeah, I'd say let's get going and see what we can find.
Let's do it.
All right, Dan, I called in that 50 meter 45, and now I called in a 50 meter north.
Roger.
So looks like we're starting to go 45 now, a little bit.
Keep it moving.
Roger.
Moving. Don't stop in to smell the flowers.
The nice grenadier in front of us. I think that's the abyssal grenadier,
Right.
Coraconortes armatus. We'll see a lot of those down here as the primary fish to observe. And one other person we have on the line is Justin Hall. He might be moderating some questions from the public tonight. Is that true, Justin?
Yeah, can you hear me coming through okay?
Yeah.
Yeah, sound good.
Oh, great. Well, we're doing a little midnight moderation because, you know, some of us are nocturnal and it's a good time to take some questions from the public for the night owls out there.
Oh, well,
Or it depends on.
yeah, I was going to say we have some European audience and people who
I was
are
just
up in
going
the
to say,
morning
yeah,
now.
or for the morning, for the early birds. So yeah, my name's Justin Hull. I work for Greater Farallones National Marine Sanctuary. We've opened up the question section on the front page of Nautilus Live, so you can type in your questions. And Chad, I know that you've got a lot of science to do, so I'm going to be very cognizant of that. Everyone's got science to do, but we'll try to get some questions in between midnight and 2 a.m.
Sounds like fun.
The fish in front of us is the giant cusk eel. It's pretty common down here. It's the largest cusk eel, hence the name, giant. And an octopus.
I know a few hours ago, people were counting the number of octopus that we're seeing so far. Has that continued or knocked off? Any rough estimates, Chad?
Oh, yeah, that was stopped a long time ago. I actually was saying earlier, I forgot I was going to throw it in my bag last week. I was going to bring a pitch counter.
Oh, yeah.
To just sit here and just mindlessly click as I saw these guys, but I forgot it. It would have just been a rough estimate anyway. But I mean, conservatively, we're well over 500 or 600. I wouldn't be surprised if we've seen over 1,000 already. I mean, some of these frames we've seen tonight have had more than 40, 50 octopus in just one frame. And then we've seen dozens and dozens of those kind of scenes. So you quickly get into the hundreds. And we got a lot more octopus to see.
And after the cruise with the video we're collecting, we will be able to count them. That's our goal, right, Jeff?
Absolutely, yeah. So at some point in the future, we'll have one of us, or perhaps you know, could be another scientist or an intern,
Add one more step north, 50 meters. Thank you.
Or someone to pour through these hours of video and count the octopus.
I imagine there would be a few people watching tonight that would sign up for that if they had the chance.
Yeah, exactly. And I know that, you know, these aren't robust transects where you have a fixed width, a fixed time, a fixed distance, so you can get density estimates. We're not looking for that. We're just looking for minimum population count, but you still have to be cognizant of not going over the same area too many times because you want to avoid double counting. We certainly haven't even come close to doing that yet because we're doing a full circle around kind of the upper edges of this cone. So we have not been over the same land twice yet.
So the species of octopus we're looking at is Mus octopus robustus. Have we seen any other species?
Not that I've seen today. No.
But in other parts of the seamount, we do see two other species, at least, of octopus. Bent octopus, which is related to this moose octopus, and also Granellodoni. But down these depths, I think we're primarily seeing the moose octopus. And Chad, correct me if I'm wrong, but this is the second place in the world that such an aggregation of this species has been observed, correct?
Yes, so they're the first aggregate. So these are common octopus. Moose octopus are cosmopolitan in most of the world's oceans at these depths. So the species itself is not rare. They've been seen brooding eggs in solitary situations all over the world. But what hadn't been seen until it was witnessed at Costa Rica is they were all clustered together in vents of warm water. And so that was brand new. And so they were thinking something about this warm water is attracting them. Maybe they're gaining an advantage of some sort. But after multiple visits with an ROV there, they never saw an egg hatch. They never saw any eggs developing. You can usually see their eye spots and arms through the egg case itself. Well, the problem is that this warm water coming out of the seafloor is inversely related to oxygen. So the higher the temperature, the lower the oxygen. And what they were finding there is that they theorized the oxygen was just too low to support development of these eggs. Basically, maybe these mothers were thinking they were doing the right thing, but they were sentencing their progeny to a certain death. But here is the second place in the world it's ever been discovered like this, about 10 kilometers away, actually. This is now the third distinct location in the world. So we have 2 of the 3 that have been discovered right here. But here we have seen not only eggs that are developing with eye spots and arms, but full-on hatching events. We've seen them come out of their egg cases right in front of our eyes. So we know that this is a viable population, or nursery, if you will. And the temperatures aren't quite as high here as they were in Costa Rica, at least not yet. Temperatures range from a few degrees Celsius to 10.5. Down in Costa Rica, they're getting 12 and 13 with lower oxygen levels. So this might be more of a Goldilocks situation where here at Davidson, they have just the right balance of oxygen to support their development, but it's also warm enough where it might be accelerating or shortening the time it takes to develop those eggs, because these deep sea octopus can take years to brood their eggs. That's the amount of time the mother's sitting on them and oxygenating them. There's another species of octopus here that Mbari studied. And it took her 54 months, 4.5 years to brood her young. And so we don't know how long this species takes to brood, but we are making the presumption that it accelerates that development. But those are a lot of questions we have, and there's going to be a lot of science hopefully in the future that's done to kind of answer those questions.
And is it thought that this is the last life event for the females, that to create these progeny and then pass on?
Yeah, just like most other octopus, this is what they do in life. They are born, they grow up, they feed, they mate, they lay their eggs, and the moment they lay their eggs, they never feed again. But they take care of oxygenating their eggs until they hatch, and then they go through a relatively slow process of senescence where they die, they kind of not dissolve, but their body breaks down, and they become kind of recycled with the environment again. But that's what, in fact, my kids, I showed them, we sat down and watched My Octopus Teacher on Netflix, which was a pretty remarkable story. It was actually really good, but my kids had no idea that this is what octopus are destined to do. It was this great story of this.
Chad,
Let's do 30 meters,
Oh, go ahead.
bearing 315.
I was just gonna say, it sounds like we have quite a few questions coming in, so whenever you are ready, we could take some.
That hesitation must be readiness, Chad.
Oh, I'm ready.
Oh, you're ready.
Bring it on.
All right, you seem pretty engaged, Chad. I know you're compressing a lot into this dive, and it's very infectious for me. One of our questions, we've got a couple people ask this, is that the octopus look a little bit vulnerable to predators in the way they're situating in these crevices. Any thoughts on that?
Yeah, great question. As far as we know, there probably aren't too many active predators of these octopus, and Erica knows a lot more about the fishes than I do, but we haven't seen any evidence of these mothers being preyed upon by, any kind of sharks, and of course down at this depth, I don't know really how much there would be down here anyway. Yeah, it does. They do look like they're vulnerable, but at the same time, that's where their suckers are and their beak and everything else. We don't know how long they're here, though. That's the interesting question. If they do take several years to develop their brood, is the warm water accelerating it to half that time, to 6 months, to, you know, those are, again, the questions that we just don't have answers to. But we have not seen any evidence of these mothers getting preyed upon. However, I would not rule that out, but it doesn't seem to be a significant problem for them, I would say.
Oh, and we have a technical question for our technical team. Someone said that they watched all of last year's season and they really are enjoying the telestrator, and they wonder if there's any other new perks to the equipment this year.
Well, the whole control van is brand new, and we have, rather than 2 vans put together, we have a third in the back for studio interactions. All the screens are new, brand new in the van. They're all widescreen now for the computer monitors, which is a definite upgrade from what they were before. A lot of new cameras.
Excellent. Great. Thanks for chiming in.
I will say that I was on the Nautilus the past 2 previous seasons when it was the older control van. And I was real excited to see this. And
Argus sonar and
when I came in this morning, site. It literally had the new van smell. Smelled like a new car.
If you take a look at the quad view or satellite 3 feed, you can see a wide shot from the back of our new van setup.
Nice.
Yeah, if you come west of it, that'll help. West, northwest.
Oh yeah, here's a question that folks might be wondering if they didn't watch our last watch. They said that you mentioned it's a nursery for the octopus mothers and the babies. But are there
Roger that. We're
males
just finishing this circle, and we're going to come around here west. So I just moved. The last move was, I think we drifted too far east, so just kind of go under Argus. We're still got a couple more north moves, and then we'll be coming around the circle, around the hilltop. I think just covering, so we just now are reaching where we were at the beginning. Yeah, that'll help. Just now reaching where we were at, so I think we're gonna start a circle, concentric circle, and with a smaller diameter inside the circle we just did.
Correct, yeah. That would be great.
And if you want, Dan, I can always put this here. I can always switch these screens if you like. Yeah.
Great. Want to make sure our navigation gets taken care of first. So yeah, the question was whether there are males in among these mothers with their babies, and if there's a size difference between the males and the females.
The males are slightly larger than the females. The females' mantle, kind of that head portion, I guess you can describe it about the size of a large grapefruit or a 12-inch softball. So they actually are a little smaller than maybe they appear on camera. I was shocked the first time I saw one that was collected. It was smaller than I had imagined, but the The males are slightly larger. So the ones that you see in this upside down posture, those are all females. Males do not exhibit that posture. That is the brooding posture. That's specific or unique to this genus, which is moose octopus. I believe there's like 13 or 14 species, and all of them do this inverted, weird-looking flower posture. Well, at some point we can point out some males. Some of them that you see not in the brooding posture could be a male, like this one coming up is probably a male. You can actually identify them. You can sex them by looking at their third arm. They have a modified arm that's shorter. It's like a club-tipped arm, and that's where they transfer sperm packets to the female into their mantle cavity, and that's how they mate. So we'll see the males kind of snooping around for females at some point, but we'll be sure to point those out, and we'll get into more dense clusters of females. You saw just a couple minutes ago there was a good line of about a dozen or more in these cracks, and that's where you see a lot of these fissures of warm water coming out of the seafloor. Right now we're seeing it's fairly sparse, but we'll run into dense aggregations at some point.
When you're trying
But
to,
isn't it true that, sorry, I just want to mention one thing. Isn't it true that where these octopus are laying, the rock seems to be darker and cleaner, and that could be caused by the warm water that's coming out, but also she may actively be cleaning it with her arms occasionally. Is that right?
Yeah, absolutely. And we know that some of these spots are visited by different females because we've seen females with already cemented eggs, and to kind of a little to the left and a little right, you'll see evidence of cement used by a previous female that was occupying that spot. These females, when they lay their eggs, they produce this oviducal cement. It's like an underwater cement they produce from a modified salivary gland, and it's green in color, and they'll just stick it on the rock.
Let's do one more step, 30 meters, 315. Thank you.
Ooh, this one we should get to sooner rather than later, Chad. Some people are wondering if they should go to sleep or if they should stay up for the whale fall, because they're kind of trying to keep their eyes open. And I feel like we should tell them.
That's a great question. Oh,
Your
man.
best guess.
I'm having that problem too, because I technically am supposed to be doing one shift, and I'm finding myself just staying up because I love this stuff and I want to make sure everything's cool and I don't want to miss anything, you know, a little FOMO. But yeah, the whale fall, best guess is the latest we would arrive there is 10 a.m. Pacific, but more than likely it will be before that, up to maybe even around 6 a.m. So I'd confidently say anywhere between 6 and 10. It really depends on how quickly we finish this goal here at the octocone. How much we stop, how much we measure at the end, and then on the transit over to the whale fall, there's a target we want to investigate. And it's one of those things where if there's something great there, we're going to spend a little time. If there isn't, we're going to go right by it. So that's kind of the best guess right now, 6 to 10 a.m. Pacific, that is.
Great, thank you, Chad.
Chad,
Go ahead, Erica, were you going to chime in?
No, go ahead.
I was just going to mention all of these incredible sponges that we've been seeing, some white, some yellow, some large. And we've also been seeing 2 species of black coral that look a bit like a palm frond. And those are incredibly long-lived species. I think even, Erica, can you tell us how long those can live?
You know, I don't know the age of the Bathopathes and Alternatipathes, if I'm saying that correctly. Those feather-like, looks like a quill or a pom-pom, like you said. I don't know how old those get. But corals down here seem to be long-lived. Some other species could get up to 200, 400 and older, 100 years old. So things down in the deep grow a little slower. Maybe because there's no, they're not falling down, they're not dying, but they just have a slower metabolism as well. So they grow slower, live longer.
Seems like things move at a different speed down here.
Yeah.
Chad, you said you can sex the octopuses by their third arm. When you're counting octopus arms, where do you start and which direction do you go?
Good question. So if you're looking at an octopus from above and the eyes are pointed at 12 o'clock, that's where you start. So the first arm to the right of that center between the two eyes is arm number 1. You go to arm number 3, and it has if it has a club-tipped shaped end to that arm, that's a heterococcylus, which is that modified arm. So when you see it, it's obvious, but often they'll have it hidden, tucked, it's behind a rock, and you're like, I can't tell.
And it's identifiable throughout their life?
Yes. Although it's a good question, actually, that you bring that up. I don't know about like hatchlings. I think that is something that, develops as they grow up. It's not from hatching. Oh, there's a large coral dead ahead.
Wow. I don't know what that is.
Oh, that's spectacular.
Yeah, if we can just take a quick gander over there and just get some screen grabs or some couple zooms, and then we can be on our way. Normally, these are the things we'd like to stop and check out for longer, but we just don't have the time.
I think it's a black coral, but wow, I've never seen that one before.
No,
I'll put
this
you on
is
STL, Dan.
this is different. Yeah.
We have it framed up in our digital stills camera that's on loan from.
Oh yeah, I see it back here.
OSU, and
just noticing our lasers are off at the moment.
Oh, thank you. Yeah, that'd be good to get measurements. So we have these lasers. You can see they're green. They're at a fixed width of 10 centimeters apart. So as you can see, when you shine them on an object, you can take those frame grabs and then do the math. How many laser dots can go across that thing? And then you can get some estimates there. That looks like from left to right, that's easily more than a meter across. But that's
this may be something we've seen before, but never this large.
No,
it's quite amazing.
Yeah, it seems like a black coral, but I usually just see them kind of the single stalks. That's like a whole, yeah, I don't know.
Is this something we might want to grab a small piece of, or not on this dive?
5 meters. Would you please?
I think we probably need to move on.
Okay.
Yeah, we'll just stick with the video right now. We just don't have too much time for collections at this point.
Dan is pretty fast at these kind of things, just saying.
I'm right at the end of my leash to the south, and Argus is to the north of me, so I'm gonna try and come around it here, but I'm probably gonna run out of leash and have to run away.
No worries. Yeah, so
come down another 3 meters, Jake.
That is just beautiful.
It is. This is pretty cool. I've never seen anything like this here.
On your way out, Dan, you can see in Argus there's a patch of octopus.
Roger, I see it.
Oh, that's a nice big pool coming up to the right.
Okay, that's as far as I can come around.
No, thank you. So one of the things
the ship moves.
Yeah, at home for
still west. Would you like me to hold it?
Nope, keep moving. That's our marching orders, right?
Roger.
Move it, move it, move it.
Yeah, there'll be a big patch of, relatively big patch of octopus coming up here in Hercules' view. In fact, it's starting to come into frame on the right side.
Unless, yeah, unless Chad wants to hold.
Well, yeah, if we can kind of just do a flyby a little closer.
Yeah,
just for counting purposes. Normally big clusters like this will stop, land, look at the shimmering, look at the community, and then take some oxygen and temperature measurements where we see the highest amount of shimmering. But again, we just don't quite have that time. But you can see right there, I mean, 1, 2 is easily over 30 right there.
Why is we're always time pressed on these dives, Chad?
What was that?
Said it seems like we're always time pressed on these.
I know last year we got here in the last hour, 2018 the last hour. Now we have a weather system coming. I know, I wish we could stay in these places a while.
Like weeks, I'd be happy to be here.
I would love to do that.
Bridge nav.
Just soak for 5 days here at the...
One more step, 50 meters west.
These are good shots.
Yeah, fantastic.
Do you see any shimmer?
Not obvious right now. Earlier today, no, there's a little bit there now.
Shimmer, yeah.
Yeah, you can see it now. For those who are watching at home, it kind of depends on your internet connection and your monitor and all that stuff like that. But there are places we've landed already today where the entire monitor is just blurry. It's just completely wavy. You could see it on a cell phone easily. And we'll stop later as we cover more ground and get to the summit. We'll probably have a little bit more time to do stuff like that, but we just got to keep moving right now. One of the things I was going to say is we haven't mentioned for a while how deep we are. We are currently at 3,089 meters, so we are almost 2 miles deep right now. And it takes 3 hours just to sink this far. And so we're attached by a cable to the vessel, and we can only go as far as that leash allows us
I'm
to.
down 3 meters.
And so what they have to do, and you'll hear it in the background there, is they
I
have
just
to
need some lace to get under you.
Yep, coming down.
they have to call to the bridge to ask them to move a certain distance in a certain direction. And it takes time for that to happen. It takes time for that motion to translate down the line. And then it takes time for the ROV to catch up. So it's like a slow motion tug of war. So the ROV can't just move any direction it wants to. It's limited by these factors. And the deeper you go, the longer it takes and the more dance it takes and the more choreography it takes to do these moves.
Yeah, Chad, I was going to say if we get to do a Channel Islands dive, you'll be spoiled with the shallow movement where everything, where Argus just moves around on a dime.
So just in case anyone at home is a little confused if we're talking about things before you're seeing them, sometimes there's a little bit of a delay where the voice gets to you before the video. So if it's confusing, just wait, be patient. It will be coming to you soon.
That's a big boy there.
Yeah.
And speaking of a little bit of a delay, we toggled the green light on for taking questions, but for some people, we're hearing that it's still red. Here's a little tip. If you type it in there, it seems to be coming through to me. So feel free to post your questions even to the red button. It's coming through.
Even if you don't have a question, tell us where you're viewing from.
We have a question coming all the way from Norway. Noah in Norway was wondering why the octopus curl their arms around their bodies like that.
So when they're upside down, we found out by distinct interviews with these. No, I'm just kidding. they're usually when we zoom into these
Great
mothers,
target shot.
it's really hard to see their eggs. So they're doing, that means they're doing a good job of protecting their eggs from predators and anything that directly get to them. Number two, the way their siphon is, that's their kind of X-current jet propulsion system will often be pointed. It's a great way to point that siphon at their eggs. And the reason for that is they'll blow water over their eggs, which oxygenates them. So there's those two main reasons there. Like we talked about earlier, they do look vulnerable, but it doesn't appear to be too much of an adverse problem for them. But, you know, they certainly aren't using their eyes to look straight up into the water column. They typically have to look out the sides. It's unusual, but it's, again, distinct to just this genus of octopus. Other species or other genus brood exactly 180 degrees the other way. They sit on top of their eggs, more like a bird.
The northern extent before we start to cover old ground.
Thank you, Noah from Norway, who happens to be my nephew and has been diligently and enthusiastically following us.
That's towards the northern extent. We covered over here on the last watch. So if you kind of turn to your port, and then
we've circumnavigated. Is that what you're saying?
We're trying to do an inner circle. So don't go any more north and just keep coming west if you can.
Right.
Or yeah, west. That's right.
I think we're seeing a bunch of stalked crinoids, and right behind it looks like a whole nother group of octopus.
Yes.
Wow, really cool.
Looks like Dr. Seuss land. We say that a lot.
Wow, is that one of those single polyp octocorals?
It's the single polyp yeah, what's the name of that one? Do you remember, Erica?
Steve will be excited
No,
about
and
that.
I saw that earlier and I was in disbelief.
Yeah.
If we, I don't know if we can get a tight shot of that. I know we might be flying over it, but.
Yeah, we can.
That is incredible. Steve A will be very pumped about that. Taylor Ann, you'll have to pass that along to him. I think it's only just been described in the last year.
Talking about the sky below me here.
Yeah, that one did. Wow.
Looks like a succulent.
Yeah.
Yeah.
It does look like a succulent.
A succulent. Suck your faciulent. Or alien-like.
Yeah, or alien. Maybe the rest of the body is down deep. That's just the tip.
Just pause here and get a let our DSC take a still photo while we're not moving, and
yeah, it's a good frame up. Nice.
we'll be able to crop it out later. Come on, Herc, stop moving.
Erica, do we have the species for this?
Tell me when it
No,
goes.
we don't.
I think we
What did
saw
you
one
call
maybe
it,
just
Chad?
a few
Steve?
minutes ago.
Quick zoom. That's good.
Wow.
You know what this might be? This might be the soft coral that's related to the mushroom soft coral called Bathy alcyon robustum.
Okay,
Maybe.
Steve, zoom out for me, please.
I'm going to call another one in. Bridge
That's what I
mount.
think it is.
One more move, 40 meters west. Thank you.
Yep, that's what it is. It's a relative of the mushroom soft coral. And it's called Bathy alcyon robustum.
I think so. Dan, do we want to keep porch light on? Steve's
What's that?
asking. Are we keeping porch light on? Steve's asking. It's normally on.
No, the porch LED should be off, thanks.
Okay.
Unless for sampling, should be off.
Good. Roger.
That was impressive.
Okay, moving west.
Yeah, it just makes it a little easier
That
to
was
expose the
impressive.
whole frame when it's not so bright.
Yeah, it
front.
totally blows it. If we leave it on, you can feel free to yell at us, Steve.
Sure, sure.
Just endless fields.
Well, what's going on here? Which way do I need to go?
Yeah, keep going west. Should be.
I'm going west, but yeah.
Chad, I don't know if you already talked about this, but
South or north or
Did
where am
you-
I in relation to Argus?
did you get
Argus
the
is looking
chance
south.
to dive in a
Ish.
submersible here?
I know that, but where am I? South of Argus? I need to come to the north.
Roger, you are south of Argus, and that's okay where you are as long. Yeah, a little bit to the north, and
I need to
keep moving
slide to
west.
the. I feel like I'm tight. I see. I get it now. Thanks.
Sorry about that. So our USBL is not helping us any.
Lost the plot there for a minute.
Yeah, very close to here. The original octopus garden about 6 miles away. The BBC, after the discovery we made in 2018, was interested in broadcasting live from here to Blue Planet Live back in the UK and Canada. And so they invited me along, and I got to do 2 dives in the Alvin, one at the octopus garden and one on the summit of Davidson Seamount. So I'd never been in the Alvin before, so that was a pretty amazing experience to see these mothers, face to face, literally.
Wow.
Or to sit under 3-meter tall bubblegum corals, because it really gives you context for size and scope and perspective, because you see a lot of these things on the TV screen, but then when you get down there, it's all in 3 dimensions, and it's a completely different experience.
That's awesome.
But this ability to send a robot down to soak in the water for more than 24 hours, to have multiple experts on shore see the live feed, there's no danger to humans. We can rotate shifts. I mean, this is definitely has the advantage over submersibles for sure, in terms of pure science observation, number of hours, all of those things. But if you have a chance to dive in a sub, it's worth it 1 or 2 times, for sure.
How much bottom time did you have in Alvin?
They were not full days. Let's see, I think the Davidson dive was 5 hours and the Octo Garden was 6 hours. But the Alvin goes up and down faster than the ROV.
Sounds like a challenge.
Yeah. Yeah, it sinks really fast. It was 40 to 45 meters a minute, I believe. And then coming up, of course, they have all that ballast weight, all of that steel or iron that they jettison so we can pop like a cork.
Hold that position right there. Thank you. So I think Argus still has a bit of west sway, but I think we'll be able to move south from here, and we'll be on the inner circle of the last one. That's all right with y'all, but we'll still get a bit of west.
Still going west?
Still going west. Actually,
I'm following the ship.
Yeah. Let me revise that.
I feel like I'm following Argus too.
Yeah. Let's see. Argus is kind of like this. You're going like that. I just stopped it. It's probably got another bit to go.
West seems downhill to me. What do I need to do?
Yeah, so that's why I want to start heading south, I think.
So I can go south?
Yeah, I'm about to call that, but Argus may be a little bit
South
west.
will be up the hill for me?
Yeah, and then we might have to, along the way, drop the operation down a bit. case.
Well, I can skirt around. If
Yeah. Depending on where Argus ends up, how far west Argus goes.
so we're in our happy spot now, right? So Argus is to the north of me, because
Yeah, that's perfect. That's kind of where we want to be. And then if we need to
copy.
back down and drop, we can. If you're at the length of it.
I see what I need to do now.
Okay.
Thank you.
Bridge nav.
Sorry, you're moving west still, Rennie?
I'm going to do a south move.
What's that?
I'm going to do a south move. Stand by, bridge.
Roger.
Yeah. I think we're farther enough west.
South move, roger.
Let's have a move 50 meters bearing south. Thank you. Concentric squares. Don't want to overshoot it and end up covering the same line again. I think we'll be able to, by the time Argus stops swinging west, I think we can manage it.
I so can't think that far ahead.
So, well, it's like a 6-minute lag or something like that, so I'm like trying to
I got my hands full just trying to.
Yeah, no, I'm trying to keep it so hopefully you don't have to move too much over there, aside from on driving.
Monitoring the chat, we're getting a lot of volunteers from an adoring public to be octopus counters.
You know, it's funny that I was just literally thinking that same thing. I was like thinking about citizen science.
Yeah.
And it would be so interesting if we did come up with a, I mean, you know, obviously one way you could do it. And what's great for the public is that all of these videos are ending up on YouTube in 1-hour segments, right? By, I think, tomorrow, but certainly in a few days. And so, you know, people can literally start looking at these videos and start counting octopus very, very soon. It'd be an interesting experiment if you could get enough people involved where, you know, maybe a small group of people each took an hour.
5 meters.
But then you have different counters.
Resetting your DVL.
There's problems with that.
That'll make a bit more sense.
Or you get enough people who would actually pour through, you know, 6 to 8 hours of video and then look at all those numbers and do a distribution plot so you can have some confidence intervals and say, okay, you know, 80% of people got X amount of octopus. That's a confident number we're going to have. That'd be interesting.
Yeah, I was thinking the same thing, Chad. I think that would be a pretty fantastic way to engage folks and come up with a really useful data.
So when you have this video and it goes out for analysis, Erica, you've done a fair amount of work in this. Does it go out to like lots of different experts?
Can you repeat the question?
Yeah, so when a video like is being captured now,
not that one,
from a cruise that gets sent out to various experts.
When I'm stretched out, you can come right down apart because we're on such a hill. Sorry, go ahead.
So like a coral expert or a sponge expert or fish expert. I know you've been part of a lot of these surveys and projects. Yeah, how do they get distributed out to these experts, and how does all that data get collected again and analyzed?
Yeah, well, typically when we're out here, we may have questions we can't answer, and then we will save the video snippets or direct them to the YouTube channel that Nautilus actually has posted. And we can ask the experts, and sometimes if we have the tissue collected with the video, that's even better, because sometimes you cannot identify things from video. You actually have to have the organism in hand. Like a sponge, you actually have to look at all the different spicule types under a microscope. Sometimes with fish, you have to count fin rays or gill rakers, which is, you just can't do that on video. So yeah, so we will hopefully tap them in while the cruise is going live, or we'll send out those video clips, imagery, and specimens to the taxonomists around the world. And I think all the specimens, they are housed, are they Harvard or URI, Chad? Harvard, right? The collection?
Yeah, I believe it's Harvard.
And maybe Taylor Ann knows that too. Taylor Ann is the data logger. She has to bottle these all up and they send them off.
Yeah, typically we send all of our specimens to Harvard. I know this year we're doing things a little bit different. But yeah, typically like last year, I know all of our samples would be sent to Harvard for identification. And this year we've been sending a lot of things to CAS, so the California Academy of Sciences. So those are our two
We're going south.
main recipients of all of these samples. And we send them to other places as well, depending on who's requesting what.
The museums have great partnerships with other museums and scientists where they can actually put them out on loan to a reputable taxonomist or scientist to do studies on them. When we were doing work at Davidson a few years ago, we collected larval fishes and we sent them to California Academy of Sciences, but can also look at old specimens from years collected earlier, decades earlier, years earlier, or even 100 years earlier. They're preserved really well, and we can compare rarely observed species to each other and say, oh yeah, this is the same one. So it's great to have these museum collections for long periods of time.
And hearing how different people bring different expertise on these dives, I could see how, you know, somebody who's looking for something really specific would see something no one else would see. Like hypothetically, maybe a chitin lost in an ocean of octopus that everybody's obsessed with, and then there's a little white chitin, and you know, nobody's interested in that chitin. Just hypothetically, in fact, they might even sort of roundly mock their colleague. Might even roundly, yet affectionately, mock their colleague who's really excited about a deep water chitin. Hypothetically, yeah, I could see that. And then, but maybe there's a community that person belongs to, the sort of molluscan experts who would bring him or her into the fold and sort of be excited for them about that sighting.
Society of chitin enthusiasts.
Exactly.
I may have been roundly mocked in the last watch for being excited about a chitin. It's still a little raw.
Shame on whoever mocked you.
So here's a great place where you can see a lot of the three-dimensional habitat that these octopus have, the caves, the cracks, the crevices. You see arms sticking out, and there's probably even several more in many places we can't see right now. But here's a great shot of kind of the complexity of this environment and how the octopus are using it. So presumably there's a lot of warm water coming out of all of these areas where these octopus are.
We have some questions about seeing shimmering water. It was from the last time we were in an area that kind of looked like this. I think we might see some shimmering water here.
We should sometimes,
close enough.
Yeah, if it's really ripping, you don't need to be still, you'll see it. Sometimes you have to kind of land the ROV, let everything settle and zoom in, and then you'll pick up the shimmering. And we can do that here, we're landing, so we can zoom into maybe a couple of these little clusters.
So just a reminder, these two green laser dots are 10 centimeters apart.
Bit of waviness on the right.
Yeah, so it's not ripping here, but there's some warm water for sure. You can see it on the right side, like Remy said.
We often see these little red shrimp in these crevice areas.
A little more.
And they may They may
That's good.
prefer this environment, but they also may be waiting for some of these hatchlings to come out and